As an animal model, we'll look at the rabbit response to an injection of insulin. Insulin reduces blood glucose in both rabbits and humans. Therefore, rabbits can be used to model humans in testing the potency of insulin preparations. There is no doubt about that. That's a predictive model. It has been valid repeatedly because it's been used for more than 60 years in testing the potency of insulin preparations. It's no longer a necessary model because non-animal assays have now been developed, but it is still a scientifically valid model.
In this episode, we're going to look at biologicals which aren't vaccines, with specific attention paid to the monoclonal antibody TGN1412 and the cosmetic treatment for wrinkles called Botox.
We deal with biological medicines.
Stephen Ingalls, Director of the National Institute for Biological Standards and Control.
Biological medicines are medicines made from cells, from tissues, and they are different from normal medicines because they're much more complicated. They're much more complex in their structure. A biological medicine would be, for example, a vaccine that's made in cells. And that would be hugely more complicated than, say, paracetamol. And because of that, it needs to be treated differently. It needs to be analysed differently. It needs to be measured differently.
And measuring biological medicine is critical. You can't just count tablets or weigh a medicine. You have to be able to measure how active it is, and to do that, you need to compare it against something of known activity, a standard. Adrian Bristow heads up the standards arm of NIBS. He gave me an example which shows, I think, just why biological standardisation is so important.
Rather than determining how much of the drug is present, we have to fall back on determining how much activity is present, and those are not necessarily the same things. And insulin is a really good example of this. I have in my archive a photograph of a lady called Hazel Davis celebrating her 100th birthday. Now, Hazel was diagnosed with type 1 diabetes in 1921. That's actually 2 years before insulin was discovered. She was kept alive in the only way that she could have been kept alive, by a very strict control of diet. But basically, she was a sick girl and she would have died. But she became one of the first group of patients to be treated with insulin in 1923 when it was discovered, and she lived for a further 80 years. Now, we know that the dose of insulin is important, but there's a second fact you need to remember, which is that the British biochemist Sanger was given the Nobel Prize for determining the structure of insulin in the 1950s. So for all that time, Hazel was being treated with a drug where the dose is absolutely critical, but science didn't even know what it was.
So how on earth did the clinicians manage to control dosages of insulin for all those years without knowing what dose they were giving?
Insulin as a drug, we know, has the effect that if you inject it into humans or to test animals, it lowers the blood sugar level. So our test system becomes determining the quantity of insulin based on how effective it is at lowering the blood sugar level in experimental animals, in this case mice. If you inject a mouse with insulin, if you put enough in, it exhibits the same kind of symptoms that a human does if you give them too much insulin. They become sleepy, they become twitchy, disorientated, confused. This is hypoglycemia. And you can quantify this by measuring the number of animals for a given dose that exhibit these symptoms. Now, of course, the problem is that this very much depends on the animals, because if you take very healthy animals, they will probably resist the effect of insulin much better than animals that are not healthy or hungry or haven't eaten recently. So this is a variable test system. And the only way that you can make a variable test system not variable is by comparing it against the effect of a standard. And that, in a nutshell, is exactly what biological standardisation is and how it works.
NIBS is both creator and guardian of the standards for 95% of the world's biological medicines and reference materials. It's a dizzying 600-strong list. Blood products like clotting agents, thrombin or factor VIII for haemophiliacs, more than 100 different hormones, from the ones used in fertility treatments to those used in doping by athletes. Then there's reference allergens like bee venom, ragwort and birch pollen, biotherapeutics such as interferon and TNF-alpha used for rheumatoid arthritis, and reagents used in work on HIV/AIDS, resources for work on CJD, and a vast number of vaccines. And it holds the UK stem cell bank. I asked Adrian Bristow what would happen if there weren't any standards.
What would happen if there weren't any standards? Well, what probably would happen is that the value of a unit for a drug like insulin, whose dose is expressed in units, the value of a unit would cease being international and would become local. So therapy in America and therapy in the UK and therapy in Japan might drift apart. And so, for instance, if you were a diabetic travelling, it is very important that you get the right number of units of insulin. But if you go to America and their insulin is a different strength to our insulin, then you could be in serious trouble.
NIBS has another very important world role. Because of their very diverse, some would say unique, range of scientific expertise, they're the first to be called when a problem arises with biological medicines. Like when 6 volunteers, trialling a new drug called TGN 1412, ended up in intensive care in March 2006.
Initial investigations into the drugs trial that made 6 men critically ill have raised questions about the future of medical tests.
The doctors said to me that it was quite serious, and my comment was, well, no one's going to die, are they? And one of the doctors said, well, at the moment it looks like 2 might.
This was what is known as a first-in-human trial. TGN 1412 had been extensively tested in the laboratory, and such immediate life-threatening complications were the very last thing anyone had anticipated.
Immediately there was a question about whether the product that was used in that trial was what it was supposed to be. Did it contain anything that shouldn't have been there? So NIBS was called in immediately to check the product, to look at it, and see if it was what it was supposed to be. And we found that it was. But then we were also called on, subsequently, to help with the investigation that was trying to figure out why the product did what it did. If the product was so dangerous, then how could we predict in the future that such an effect might happen with another medicine? And that was something that our scientists were able to help with.
And those scientists are in the biotherapeutics group at NIBS. I met them in their lab, and I asked Robin Thorpe, head of the group, why they got involved.
Everybody thought at first that either the antibody was the wrong antibody, or it was the wrong concentration, or it was contaminated with something like endotoxin, or it had been made incorrectly and wasn't fit for purpose. And we were called in to look at that, first of all.
And what did you find?
We found it was absolutely fit for purpose. It wasn't contaminated with anything that could have caused these adverse reactions. It was the right antibody, it was the right concentration.
I walked in and I nearly fainted. He's a young, gorgeous, hunky, 28, almost 29, muscly, hunky guy. And I walked in and he's looking puffed out like the Elephant Man, and he looks like a 45-year-old man who's had a cardiac arrest. He's got all the things all over his chest. It's his lungs, his heart, his kidneys are being supported. You know, all these vital organs have just been attacked by this drug that attacked his immune system.
Clearly something else was going on, and that's where the real detective work started. The drug was what it was supposed to be. No danger flags had been raised during its extensive previous lab and animal testing, yet in the human volunteers it caused a massive response called a cytokine storm, in which the immune system is unleashed on the body with devastating effects. Richard Stebbins was the next person to get involved.
Once we knew it wasn't a contaminant and it was a biological property of the TGN 1412, that's when I came in and was asked to have a look and see if we could figure out what was going on. Because in conventional assays, it was still inert. So we needed to develop some new assays that could reproduce what happened in the trial volunteers.
And what did those involve?
Well, that involved me using a hairdryer to dry this antibody onto plates and then add the human cells to it. And then miraculously, we got the same cytokine storm that was seen in the trial volunteers.
And presumably the drug company that had made it would never have thought of doing the hairdryer test.
That's correct. That was something we used here. The TGN antibody is a Y-shaped molecule, and the Y shape recognises a target molecule on the surface that it was designed to recognise. The other end of it is designed to interact with cells, and we're trying to reproduce that in the lab. And the best way we could think of doing it was to immobilise it, and in this case we used a hairdryer to stick it down onto the plastic surface, sort of replicate what would happen inside the body.
The results of this work were published really on a world stage because the whole world was fascinated by what had happened. And of course it was critically important to drug companies to know what had happened in this instance.
Yes, that's right. The kind of assays that we developed for looking at TGN 1412 are applicable to other monoclonal antibodies of this type, and indeed other immunostimulatory biologicals. And the pharmaceutical industry is very interested in using these to screen a whole range of different potential biotherapeutics. So I think our investigations are kind of much more far-reaching than just TGN 1412.
The new drug, known as TGN 1412, was developed by German company Tegenero to treat rheumatoid arthritis, multiple sclerosis and leukemia. It's an antibody specifically designed to target the cells of the immune system. It was being tested for the first time in humans, what's known as a phase 1 trial, simply to find out if it was safe to use in man. As time went on, it became clear that the volunteers were suffering from a massive overstimulation of their immune systems, what's known as a cytokine storm. The clinical effects of this new biological medicine were not unlike what the intensive care staff were used to seeing in cases of severe septicemia or blood poisoning, kidney failure, respiratory failure, and defects in the normal blood clotting system. Despite their grave condition, all 6 of the volunteers ultimately survived, thanks mainly to the specialist intensive care that was so close to hand. But we've gathered that of the 20 or so independent research units in the UK, only about 7 are based on hospital sites. This is an issue tackled head-on in a report from an independent expert scientific group set up by the UK government to learn from the Par-Excel clinical trials incident. The final report is due in November, but an interim report was published in July this year and makes 22 recommendations. Number 12 talks of access to emergency facilities and pre-arranged availability of ICU facilities when testing higher-risk medicines for the first time in man. This idea of higher-risk medicines keeps popping up throughout the report. The thinking is that biologically active molecules, like TGN 1412, represent the brave new world of medicine, but they do need to be tested with extra caution. So is it that the current systems for testing drugs in man, which seem to have been followed in the Tegenero case, are just no longer up to the job? Gordon Duff, Professor of Molecular Medicine at the University of Sheffield School of Medicine, chaired the group.
Well, I think the fact that TGN 1412 was a monoclonal antibody did focus to the risks of these new innovative medicines. And there's no doubt that we have several of such medicines already in clinical practice, and they've been extremely successful, transformed the outcome of many diseases. And it's equally certain that we'll see possibly a large number of developments of innovative type biological medicines in the near future. So it was quite timely that this expert scientific group was set up and got to work on reviewing the way that clinical trials are planned and authorized for these innovative new products.
One point on which Professor Duff's report sticks its neck out is the issue of how to work out a safe starting dose for testing new higher-risk drugs in man. TGN 1412 had already been tested in rats, rabbits and monkeys, and there were no signs, even at high doses, of the catastrophic effects to come in humans. And that's the point. The newer biological medicines often have very different effects in different species. So, could the dose have been wrong? Gordon Duff.
I think the dose was undoubtedly too high, and that's why we saw all 6 recipients becoming ill so quickly and so severely ill. What that tells us is that the methods used in the preclinical development and the phase of clinical transition to calculate the dose failed to predict a safe dose. And those methods were largely based on the current norms, the regulatory guidelines and requirements that were operating at the time, which evolved from several decades of very successful drug development with small chemical entities.
Professor Duff's report recommends a much broader approach to working out the first dose, including, for example, laboratory studies on human cells, and based on the minimum dose at which any biological effects are seen in animals. If Tegenero had used this approach, the dose given to the volunteers would have been 20 times smaller. Andre McLean, former professor of toxicology at University College London, is a consultant to another contract research organisation called Richmond Pharmacology, based in London. Having now studied the laboratory information that was available to the researchers before TGN 1412 was tested in humans, he feels that the risks of a cytokine storm were clear.
That risk was mentioned in the investigators' brochure, which is on the web. They knew perfectly well that this was the risk of that antibody. It was just a question of the dose and individual variation, of course. But all the information was there. The effect on white blood cells in the test tube, how the antibody behaves when it's injected into animals, so that all the signs were there, but they were not taken into account nearly as much as the highly standardized procedure which is recommended by the United States Food and Drug Administration of finding the maximum tolerated dose in the animals and then dividing by a factor, a rather mechanical procedure, highly suited, I suppose, to ticking a box. But I think that in this instance, it led people astray, that they thought, well, if monkeys can tolerate this big dose, we divide the tolerated dose by 500, and that gives them a huge margin of safety. And left to one side the information which had come from the immunologists and the pharmacologists.
With these novel drugs, researchers will clearly have to start thinking outside the tick box. And given that there was no hint of any life-threatening effects when TGN 1412 was tested in monkeys, does this now mean that animal testing has become an irrelevance, even a dangerous red herring? Gordon Duff.
It certainly doesn't reduce the need for animal testing. But in using animal testing in preclinical drug development, there will be a need to perform general toxicological testing and so on. And when we go beyond that to try to understand the mechanism of action of the new medicine, I think we have to validate what the predictive value is going to be in the animal model. We have to know the limits of the usefulness of the information that can be derived from animal experiments. We're not saying at all that animal experiments are not valuable. We just need to know how to get the most from them.
And how to interpret them properly.
And where their limits are when it comes to predicting a human response.
This last point wouldn't have been unusual to hear 100 years ago. Some scientists say you can never know the human response until you actually test a drug or chemical in man. I've referred to these scientists as scientific anti-vivisectionists throughout this work. However, there are scientists, from Claude Bernard 100 years ago to Professor Gordon Duff just now, who cling to this idea that if they could just work out some unspecified missing ingredient, everything would then fit neatly into place and animal tests would become predictive of human response. This is the great error stroke myth of nearly all the world's toxicologists. Let me play you an extract from another BBC Radio 4 programme I first used about halfway through this work.
It moves from the company's official discovery stage to the development stage. Years more work costing very much larger sums of money. This development stage is when a drug takes on more than a laboratory reality. Test tubes are all right, but what'll it behave like in a living body? But before any new chemical compound ever gets to human beings, it has to be tested for safety on animals. What's emerging from all this testing is a viable candidate chemical compound for the company to start putting its hopes on. A real HIV/AIDS drug which may do what the researchers require of it. By October 2001, the drug is ready to be tried out for safety in a few healthy human volunteers for the first time. This is phase 1 of the development process.
We would start off with a dose that usually is 1/100th of the dose at which you started seeing side effects in animals. So because you start at such slow doses, the average length of a first in-human study is actually about 3 months long. The safer the compound, the longer the study.
Now, we know that a lot of drug candidates fail to get anywhere near the marketplace, and I assume that quite a lot of them fail at the phase 1 test, don't they?
Oh, absolutely. I've been involved with drugs where after a single dose we stopped the trial. We would stop a drug about 40% of the time.
40% of the time? That's 4 out of 10 you would stop?
That's correct. If we're doing our job well, that's how frequently that we should be doing it.
If animal tests were predictive of human outcome, how is it that 40% of them fail at phase 1 clinical trials? Furthermore, we have no idea how many good drugs have ended up in the pharmaceutical dustbin because they reacted badly in animals but might have been perfectly safe for human beings. This is why more and more scientific anti-vivisectionists are asking for the animal testing stage of drug development to be taken out of the equation. They maintain that only human data should be in the equation. This, they argue, will make toxicology a much more predictive mechanism, and thereby make it much more scientific, and thereby make it much safer for humans to take these drugs, especially in phase 1 clinical trials.